All right, good evening, everyone. How's everybody doing? Hey, hey, hey. Welcome to Friends of the Columbia Gorge's webinar, Under the Surface, the Hidden World of Gorge Salamanders. We are very thrilled to bring back our wildlife webinars just before summer to focus on one of the most interesting spring animals in the gorge, salamanders. We appreciate you all attending tonight, and we are going to have such an amazing journey into the world of herpetology, which is the study of reptiles and amphibians. And we have two incredible experts in the field, so you really are in for a treat tonight. So while you are getting settled and we wait for a few more to join us, I just wanted to go over some Zoom housekeeping before we get started. So tonight you'll notice that your microphone is muted, and that is because with so many of us here together, it's best to give everyone the best chance to hear the speakers without each other's background noise. Your cameras are also turned off so we can pay full attention to the speakers. Throughout this webinar, there are two ways to interact with us, either through the Q&A or the chat box. We will hold all questions until the end of the webinar to ensure we have enough time to get through everything. After the last speaker, we'll go straight into Q&A for about 15 to 20 minutes, facilitated by me. And if you have a question for the speakers, which we really encourage you all, please type it into the Q&A box located at the toolbar on the bottom of your computer, tablet, or phone screen. If you're using a tablet or phone, please touch your screen because the toolbar should pop up at the bottom of your screen, but sometimes, hey, you never know, it'll be at the top, to the side, so just take a look. And then we will do our best with the limited time that we have to get through all of your questions. But if I can say anything, is that tonight is a night to nerd out, especially since salamanders are so active right now this spring. Okay, and then another thing is this webinar is being recorded and the video will be uploaded to our YouTube channel tomorrow. So if you want to go back and watch this webinar or our previous gorgeous wildlife webinars, you can go back and watch those, or you can share with your friends and your family. And you can check out our webinar playlist at gorgefriends.org/webinars. And honestly, while you're there, subscribe to our YouTube channel, give us some love, and you will also stay up to date with our latest videos. Okay, so that wraps up our Zoom housekeeping. And so let's get to know a little bit about us. Again, I want to thank you so much for being here tonight, and we're going to learn about salamanders that can be found in the gorge. Not only are they cute, but they also play a super important part and role in our ecosystems. So we will get into that tonight. My name is Melissa Gonzalez, and I am your host for tonight. I am the Public Engagement Manager at Friends of the Columbia Gorge, and our presenters for the night are Kenzie Hammond and Christopher Cousins. Our first speaker will be Kenzie. She is Friends' Youth and Community Education Specialist. She's a herpetologist, an adjunct professor of herpetology and environmental science at Clark College. What can't she do? Kenzie received her master's degree from Oregon State University, where she studied parental care and poison dart frogs. Kenzie has a deep love for all amphibians, especially those in the Pacific Northwest. And from a young age, she has really honed in her herping skills and was able to identify every local amphibian by the age of 8. Could you do that? I don't think I could do that. And so now, full circle, she teaches the reptiles and amphibians of the Pacific Northwest course at Clark College. Our second speaker will be Christopher Cousins, a PhD candidate in the Fisheries, Wildlife, and Conservation Sciences Department at Oregon State University, where his research focuses on the torrent salamanders, a family of salamanders endemic to the Pacific Northwest. This ancient family has 4 species in one genus, and 2 of those species are currently being considered for listing under the Federal Endangered Species Act. The Columbia River Gorge is a stronghold for the species and one of Christopher's areas of research focus. So before I pass the virtual microphone to Kenzie, of course, I got to tell you a little bit about Friends. If you are new to our events, I would not be doing my job if I didn't plug ourselves. Okay, So a little bit about Friends is we are the only conservation group entirely dedicated to protecting, preserving, and stewarding the gorge. Friends led the fight to protect the gorge by helping create the Columbia River Gorge National Scenic Area over 40 years ago. And we've been working ever since to safeguard the gorge and make sure the natural wonders you enjoy today will be preserved for future generations. Friends is entirely dedicated to protecting and enhancing the scenic, natural, cultural, and recreational resources in the gorge. And lastly, if you would like to check out any of our free guided outings this May or June, please come out with us. We have incredible leaders. And if this interests you, please visit gorgefriends.org/outings to learn more. Okay, well, enough of my voice. I'm gonna go ahead and pass it off to Kenzie now. Kenz, take it away.
Hello, everyone. Thank you so much for attending this webinar. And Melissa, thank you for being the host tonight. It is so nice to be able to step into the speaker's shoes about a topic that I'm so passionate about. Here at Friends of Columbia Gorge, I've been able to share my love for salamanders in our youth programs, teaching students how to respect salamanders in the environment and teaching them where they can find salamanders in their own backyard. I'm so fortunate to have grown up in the Pacific Northwest, and from a young age, as Melissa said, I loved identifying salamanders and frogs and going out with my family. So a lot of my knowledge really came from a young age and just studying and researching and reading every book I could get my hands on. So it's really been full circle to be able to make a career in educating others about salamanders in the gorge. So today in my presentation, I'm going to give you a brief snippet of the full lecture that I give at Clark College, which is all about teaching identification skills to students. So tonight you're going to be learning about the 5 families of salamanders that we can find in the Columbia Gorge and 12 specific species. You're going to learn some basic identification skills and about each species' specific habitat needs and where you might be able to find them. At the end of the presentation, I have a little quiz, so make sure to take some mental notes or some written notes, and we can test your knowledge to see how many salamanders you're able to identify after tonight's presentation. So the first family of salamanders we're going to talk about is newts. We have one local species of newt in the Columbia Gorge pictured here, which we'll go over on the next slide. Newts are unique in the fact that they have aquatic larval stage and terrestrial or aquatic larval stage and aquatic breeding, but live in a terrestrial adult life. The newts in our area have a rough skin, which helps you be able to identify them from the salamanders in our area. Newts across the country don't always have rough skin, they sometimes have smooth skin. All newt species are poisonous, containing toxic glands within their skin. The first species I'm going to highlight is the rough-skinned newt. This is kind of my gateway species into falling in love with herpetology. From a young age, I would go and catch these guys and get to hang out with them out in the wild. And they are so charismatic and they're beautiful, and you can see them in a lot of places all around the Columbia Gorge and the Pacific Northwest this time of year. They are up to 8 inches in total length from snout to the tip of the tail. They have a rough, dark brown skin pictured here and a bright orange underbelly on the underside. And a fun fact about them is when they are feeling threatened, they will actually arch their back up and their tail up to show predators that they are indeed toxic. They have enough toxin in them to kill an adult human. And in non-breeding season, they can be found under logs, and they travel long distances back to ponds for breeding season. If any of you have seen them in the wild before, you will probably have seen huge piles of them because during breeding, they will actually get into what's called a mating ball, where a bunch of them are competing for the female's attention in order to breed with her successfully. And a fun fact about them is garter snakes and rough-skinned newts have been in an evolution arms race. As newts get more toxic, garter snakes get more resistant. Garter snakes are one of the few animals that are able to eat rough-skinned newts without getting affected by their toxin. So that's something you can learn more about if you're interested in this field, but I will leave it at that for rough-skinned newts. The next family of salamanders we have in the Columbia Gorge are mole salamanders. We have 3 unique species in the Columbia Gorge. Adults in this family are typically large-bodied, and their larvae are aquatic with long gills, and as adults, they have large, well-developed limbs. They can live either an aquatic or fossorial or underground lifestyle, depending on the species, so there's not a one-size-fits-all approach for this family. The first species I'm going to highlight is the long-toed salamander. They are up to 6 inches in total length from their snout to the tip of their tail, and they have long toes, which pictured here I'm going to show with my mouse, that make them unique from other salamanders. So when you're looking for salamanders, if you find a salamander with long toes like this, that's usually a pretty easy identification marker for these salamanders. They have a brownish black body, and they have a yellow jagged stripe down their back. They have usually white speckling along their belly. And from my experience finding these salamanders, the color can vary quite a bit from this yellowish color to a really muted olive green color. So color is not always the best identification factor because there's quite a bit of variation from species to species. These salamanders spend most of their life underground. They have an aquatic larval stage, and we have 3 subspecies of the long-toed salamander in the Pacific Northwest and in Oregon. The next mole salamander we have is the blotched tiger salamander. While this species is not super common in the Columbia Gorge, and it is unknown if it was introduced to eastern parts of the gorge or if it's within its native range, they are quite large, up to 13 inches in total length from snout to tail. You can identify them by their olive and yellow coloration with black splotches covering their body. So you can see they're quite marbled with this yellowish olive green color and they're black all around. They are super cute when you get to find them. It's always a special treat. They live in grassland and shrub steppe habitats, and they spend most of their time on land in burrows. Like a lot of salamanders we're going to learn about tonight, underground is a place that they thrive because it is moist and it is a regular temperature for them. Our next species of mole salamander is the northwestern salamander. They can grow up to 10 inches in total length from their snout to their tail. They have dark brown to black in coloration. They're pretty uniform in color. There might be a little bit lighter on their belly, but they're pretty much all one color. They have deep grooves along their sides. You can see here, they almost look like an earthworm in the grooves that they have down along their body. They live in moist forests, and they're rarely seen due to living underground, like a lot of other salamander species. They require log crevices or rodent burrows for protection. So if you get to see one of these, you're typically going to see them on a rainy day when they're out on the forest floor during mating season, or when you find them under a log. Our next group of salamanders family we're going to talk about are the Pacific giant salamanders. We have 2 species found in the Columbia Gorge, and they're closely related to the mole salamander family. In some studies, they are grouped together in one, but generally it's accepted as 2 separate families at this time. Pacific giant salamander family is endemic to the Pacific Northwest, with ranges going down towards California and up to Idaho. They're large-bodied, they're the largest salamander found in the gorge, and their larval stage lasts longer than a year, which is not typical to all salamanders. The first one I want to talk about, I like to nickname this one the Pacific Northwest axolotl, because it's typically paedomorphic, just like axolotls are, meaning that they don't fully metamorphosize into terrestrial adults. Terrestrial adults are possible with the Cope's giant salamander, but it is uncommon and it's not typically seen. They live in permanent, cold, fast-moving streams with lots of rocks that they can hide under and feel protection from. And they're the most common giant salamander you can find in the gorge, especially around Waterfall Alley. The next giant salamander is the coastal giant salamander. This is the largest salamander you can find in the Pacific Northwest, as well as in the Columbia Gorge. The largest one is up to 14 inches in total length, so they're quite large. Adults are marbled, tan, and brown. Unlike the tiger salamander, which I mentioned a few slides back, these ones are more of a really light chocolatey brown with dark brown splotches, while the tiger salamander has that olivey yellow-green color to the black to gray splotches. So these are the two fully splotched species you can find in the Columbia Gorge, so they're pretty distinct from one another and they have very different habitat needs. They live in moist forests near cold streams, and they spend most of their life in burrows like a lot of the other salamanders. These ones are not as common in the gorge, but you can still find them. Our next family, which is my personal favorite family of salamanders, are the lungless salamanders. We have 5 species in the gorge. They have thin, long bodies with short limbs. They lack lungs, hint the names. So they have to have this really long body shape so that way they have increased surface area for respiration through their skin to occur. They have a slit on their snout called nasolabial grooves, which I'll show you in a future picture, that is used for chemosensory, which chemosensory is just a way for an animal to sense the environment, to feel sense, things like that. We have many local species, and most of them have entirely terrestrial direct development, meaning that their larval stage does not require having water like a lot of other salamanders, a lot of other amphibians in general. They are able to hatch as miniature adults and go right on into their life after hatching. So this time of year when I go herping, I can find quite a few 1 to 2 inch, really small little salamanders, and they're always a treat when you get to find them. So the first one I want to highlight, and this is the one I have found personally the most when I go looking for salamanders. This is the Western redback salamander. They grow to just over 4 inches in length. They have a thick dorsal stripe from their head all the way to their tip of their tail. The color can vary from this kind of orangish color to tan to red to yellow. And these salamanders also have what's called an axanthic coloration, where their body is almost entirely black and you can barely see this dorsal stripe, which makes identification quite difficult at times. They live in humid coniferous forests with mild winters under logs, rocks, etc. And in cases of dry weather, they can move further underground in order to regulate their moisture content within their skin. So one thing to note about lungless salamanders in general is they're very snake-like in appearance when you first spot them, and they're quite small. Their legs are very small compared to their body, so it's pretty easy to identify what type of salamander you're looking at just based off the shape of its body. And I'm going to come back to this slide in a few so I can show you the comparison between 3 salamanders that we have in the gorge that look quite similar to one another and can be kind of tricky to identify. The next lungless salamander is the Oregon encentina. They are just over 4 inches in total length. And you might notice they kind of look like the rough-skinned newt. They have a very similar coloration to them, and this is due to mimicry as hypothesized, so that way they get protection from the fact that rough-skinned newts are so toxic. They look similar to them, so that way they also get protection by having similar coloration. They have this typically pretty orange underbelly, but it can also vary to a kind of tannish, brownish color, and the top is typically pretty dark brown. They live in humid forests with woody debris. They're quite commonly found. They hide under logs, wood piles, etc. for protection. And a fun fact about the encentina is there's so many different subspecies that they look like entirely different animals. So in California, there's like over 10 different color locales that look completely different from one another. They vary from being yellow and black splotched to brown with orange. So many colors that you can see them in, and it's evolution happening in real time in California for this species as they have broken apart from one another and started to diverge from the original species. So that's something that is really interesting if you are into this that you can look further into. There's quite a few papers about this as well. The next salamander is the dun salamander, which this one looks pretty similar to the western redback salamander a lot of times. They grow up to 6 inches in total length. They're brown bodied with a tan or green dorsal stripe, and the color of stripe is present on the sides in the spots. So I'm highlighting here, you can see this dorsal stripe on this dun salamander is a pretty yellow color, and on the side you can see the spots in the yellow color down here. So for the western redback salamander, they also have spots. This one doesn't have very many, but it won't be the same color that is on the dorsal stripe. It'll be a lighter white or blue color. So that is one easy way you can tell apart these two salamanders. These ones can get quite reddish in color too, just like the western redback. And another note about them is their dorsal stripe doesn't extend all the way to the tip of their tail, just like the western redback salamander does. They live primarily under rocks near splash zones and streams, but they occasionally can be found under logs as well. Next, we have the Oregon slender salamander. They are up to 4 inches in total length. They have a red colored jagged dorsal stripe and marbled white and brown belly. So you can see their underside is quite white. This one's kind of curled up here in this picture. And the color on the top is quite vivid and bright in person for the ones I've been able to find, is they're a pretty red to orangish color. They live in moist old growth forests under decaying logs, and they also can be found in moist talus fields and lava fields. So they have quite a bit of variation in the kind of habitats that they can be found in. Oregon slender salamanders also are unique in the fact that they sometimes can be found in groups to stay damp. So when you flip a log, sometimes there'll be multiple of them, so that way they're huddling together in order to keep their moisture amongst themselves. And the last lung-less salamander I'm going to mention today, there's countless others you can find in the Pacific Northwest, but it's our large mountain salamander, which is actually endemic to areas surrounding the Columbia Gorge. If you have followed our magazine, I featured the large mountain salamander in the wildlife feature of our article because they are one of the only endemic species of animal to the Columbia Gorge. They're up to 4 inches in total length. They have a yellow or tan dorsal stripe that extends from the base of the head, you can see here, all the way to the tip of the tail. They have a pretty scalloped edge along its dorsal stripe. And the one way you can identify them apart from the western redback is on their underside. They actually have a pinkish to red coloration along their belly. They live in shaded, steep basalt talus slopes, so this habitat is a little bit more unique than the other salamanders. So they really thrive in those talus rocky environments that have lots of moss growing on them, lots of moisture retention, and great temperature regulation. So a similar habitat to the pika that we all know and love, but a little bit more shaded than the ones that we find the pikas in. Moving on to the last group of salamanders, we have our torrent salamanders. I'm not going to go too in depth on these because Christopher's entire research has been focused around these, but we have 4 species of torrent salamanders, all endemic to the Pacific Northwest, with 1 species found in the Columbia Gorge, the Cascade Torrent Salamander. They have highly reduced lungs, and they live a semi-aquatic to aquatic lifestyle. Our Cascade Torrent Salamander, pictured here, are up to 4 inches in total length. They have a brownish coloration with bright yellow belly and white and gray speckling along their sides. They spend their lives near fast-moving streams and waterfall splash zones, so you can find them quite a bit around Waterfall Alley in the Columbia Gorge. So we're going to have a little challenge now to test your knowledge. So in your chat with whoever's watching with you, figure out if you can determine what these 3 species are. So write down what you think number 1 is, number 2, and number 3, and I will watch the chat and give out some guesses. There's no wrong guesses when you're learning, you guys. We have a few for number 2 as the rough-skinned newt and the encentina. I had one person guess that the first one is a lungless salamander, which is correct. But what type of lungless salamander is it? Is it the dun salamander, the western redback, the large mountain, Oregon slender? Have any guesses? All right, there we go. I see quite a few. Number 1 was guessed as the redback salamander. That is indeed correct. As you can see, his dorsal stripe starts at his head and goes all the way to the base of the tail. Oh, apologies. All the way to the base of the tail, and there's some white and gray speckling along the sides. If it was a dun salamander, this stripe would not extend all the way to the tip of the tail. And if it was a large mountain salamander, it would have a more jagged striped edge and a red underbelly. For number 2, a lot of you were correct. I had a few guesses for rough-skinned newt and a couple for encentina. So the difference between a rough-skinned newt and an encentina is that the rough-skinned newt has some pretty rough skin, hence its name. So you'll actually be able to see the texture of the skin in a rough-skinned newt. And in person, if you were to spot them, the rough-skinned newt is quite a bit larger than the encentina, with it being about 4 inches longer in total length. The bright orange color is the same for both. So rough-skinned newt was a great guess because they look very similar, but it is an encentina, so great job. Now number 3, I don't have any correct guesses for number 3. I will give you a hint that it is not a lungless salamander, it is a mole salamander. And to pay attention to its feet. It's not as visible here, but if you can take a look at, oh, you can't really quite see it. I have a hint though, it has to do with its toes. Yes, Roberta, it is a long-toed salamander. Great job. Thank you all for this quick quiz. If you want to practice this in the future, I highly recommend going to the Oregon Department of Fish and Wildlife or Washington Department of Fish and Wildlife's website, and you can save the photos of the salamanders and make yourself little flashcards. That's how I was able to learn how to identify so many salamanders within the gorge. To wrap up my presentation, thank you all so much for listening about ID, is how to herp responsibly. From this webinar today, my hope is that I inspire a lot of you to want to go herping because, in my opinion, it is the superior hobby to birding because you get to see critters that not everyone gets to see. So some things I wanted to point out to you all is because salamanders live in environments such as under logs or under rocks, it is important that we leave no trace and minimize the damage to the environment that we do as much as possible when we are looking for salamanders. So what I teach my students when I go out to the field is to stay on the trail while you are hiking, unless you're doing field work, of course, then you have to go off trail, or if you're on your own property. But while on a hike, stay on the trail, and any logs or rocks that you see that are small enough for you to pick up on your own, you can gently lift them up and look underneath to see what's on the underside. Sometimes you might not find salamanders. There might be scorpions or roly-polies or worms or centipedes. All sorts of cool things live under these rocks and logs. But looking underneath really gently, and if you do see a salamander and you want to get a closer look, make sure you have very clean hands. No bug spray, no sunscreen, no chemicals, lotions of any time on your hands. And if you're worried about it, using nitrile gloves that are safe for animals are a great way you can gently pick them up and keep them low to the ground so that way if they do run off, they're not going to be hurt. Take a good look at them and always release them back to the environment. We want to release them where we found them at. And what I like to do as my best practice is put the log or rock back how I found it. Make sure nothing's been disturbed. If any moss got moved, put it back and release the salamander right back next to the log or rock that you found it. Because that way, when you're gently putting the rock back down, you're not accidentally risking crushing the salamander. If handling amphibians, it's just really important to make sure that you don't have any chemicals on your hand and that you're really gentle with them, gently cupping them and making sure no harm is caused to them. If you do find salamanders, I would love to see your pictures. You can email me at kenzie@gorgefriends.org and I can help you ID them after this webinar. With that said, that is my presentation. Thank you all so much for listening. I'm going to pass it off to Christopher where he can get more in depth about his research with Cascade Torrent Salamanders.
Okay. Hi. Good. Is that look good? Okay. Cool. Hi. Yeah. I'm Chris. I'm a PhD candidate at Oregon State, in Tiffany Garcia's lab. And just want to shout out to all the co-authors. A lot of people with the Forest Service and USGS and other profs as well. So yeah, today I'll be talking a little bit about some of my thesis research. My thesis is largely focused on torrent salamanders, and with kind of some of the work I've been doing, we've found some really interesting stuff, specifically about the Columbia River Gorge. So I'm excited to share that with you. So torrent salamanders, and Kenzie already told you a little bit about them, but they're headwater specialists. So headwaters are the kind of the source of all downstream water. So like on this figure on the left, everything that's marked with a 1 would be a headwater. So this typically is like higher elevation. The gorge is a great example of how to see that kind of accessibly, because you can hike to them rather easily. And of all the rivers, like throughout the world, 80% of rivers are headwater streams. So they actually make up a very large percentage of streams. And they're often a lot more tied to their environment than some of the larger streams. So all 4 of these are different headwater streams that we sampled for torrent salamanders. So you can see that there's kind of a lot of variety in there. And one of the things that makes headwater streams so kind of distinctive and different from the larger order streams like the Columbia and others that you might be used to, is that they're very tied to the forest. So, I mean, looking at these, you can see how narrow they are, how shallow they are. And that means that the animals that live in these streams are much more closely tied to terrestrial habitats than some of the other ones. So, for example, torrent salamanders are prey items not only for owls, like barred owls eat torrent salamanders, but also spotted skunks and some things that you wouldn't maybe associate as being aquatic predators. So they're kind of an important link between that kind of forest terrestrial habitat and then the stream habitat. And I'm just checking the chat to make sure everything's cool. Okay, yeah. And there we go. They also live in some very interesting headwaters. So this is in the Columbia River Gorge. This is kind of near Wyeth. And so this looks like a dry stream bed, but there is water moving through it under the surface. And we actually did find torrent salamanders just upstream of this where the water came. So torrents live in these kind of streams, which are known as ephemeral or intermittent. And the way that they're able to do that is they're able to utilize the hyporheic zone. So kind of what the hyporheic zone is, essentially, you can see in this figure, you have the water kind of floating over, and then there's subsurface movement of water. So within all of these kind of little cracks and crevices, torrent salamanders are able to be found in there. So this is kind of a good example of what that looks like. And oftentimes in headwater streams, it may appear that some sections don't have water, but there is water under the surface. And it's really cool that they're able to use it. And actually the way they found them is we use piezometers, which are essentially tubes that you stick into the ground and you can kind of see how far down the water is. And by pulling them up, they found some torrent salamanders in one, and they were like, oh, I guess they use this habitat. It's pretty cool. And they have some kind of interesting morphological adaptations to headwater streams. So this is a larval torrent salamander. And one of the first things that you notice, or I guess don't notice, is how small the gills are. So unlike something like one of the ambistomas or the mole salamanders, where they have these really big gill structures, and that's how they breathe and take oxygen from the water, they live in these kind of slower moving, maybe wetlandy or pond type habitats, whereas torrents on the left with these very tiny gills don't have to invest in gill structures because they're living in streams where the water is really cold, so it supports more oxygen, and it's also really fast. So you really have to zoom in quite a bit to see the gills on them. And so this is just a macro photo of the gills of one of these larval torrents. And so you can see kind of the blood vessels, and that's where the exchange happens between the water, the oxygen kind of moves into their gills, and that's how they're able to breathe when they're larvae. So they're really dependent on these kinds of streams. Their life cycle is also very dependent on the availability of water year round. It's thought that their eggs can take up to a year, which is very, very long for amphibian eggs to develop, and they need to be in a cool, moist place that whole time. This is, I think it's the only like photographed Cascade Torrent Salamander egg mass. Generally, they're hard to find because they kind of go down into that kind of cobble and lay eggs in there. And kind of what lets you know too that there's not a lot of this habitat available is that, you know, the average like torrent salamander female is only going to lay about 7 eggs, but yet whenever people have found eggs, it's always been in groups of 40 or 50, which means that they're doing this communally. So a lot of this females would go to the same spot to lay eggs. And then that larval phase can take up to 6 years, depending on the thing. I think for Cascade Torrents, 4 is kind of the longest. And so within that 4-year period, they're essentially confined to water. They can't leave the water and be able to breathe. And then when they transform into adults, they're still very sensitive to desiccation and drying out, even among other amphibians and salamanders. And if the water temperature or the ambient temperature around them kind of spikes in any way, usually that's fatal. They're very sensitive to temperature, which is one of the reasons why when you handle them, you really don't want to do it very long, because just the heat from your hands can be somewhat harmful. So generally, like, I put my hands in the water when I have to handle them for samples and stuff. And it's thought that these terrestrial adults really only kind of stick to this kind of seepy habitat, except for the winter months. You know, our winter months, you get those big rain events, and that's generally when the adults are thought to disperse. So as Kenzie mentioned, there's one genus. It's endemic to the Pacific Northwest. There's 4 species. There's the Olympic torrent, the Olympic Peninsula, the Columbia torrent, and the red is on the Coast Range of Oregon and Washington. The Cascade Torrent in the blue, as you can see, is in the Columbia River Gorge. And then the yellow is the Southern Torrent, which goes down into Northern California. And 2 of them, the Cascade Torrent and the Columbia Torrent, are currently being considered for listing under the Federal Endangered Species Act. So the U.S. Fish and Wildlife Service is going through and making the species status assessment and seeing if they warrant listing as an endangered species. And the reason they're endangered is primarily due to kind of characteristics of their habitat. So as I mentioned, these headwater streams kind of face unique threats. One of that is, you know, as climate change is kind of changing, not just temperature, but how often precipitation is happening. You know, generally the kind of understood projection for climate into the future is that in the Pacific Northwest, we're actually going to be getting around the same amount of rainfall. However, instead of being spread out, it's going to be much more concentrated in these kind of localized rain events. And essentially what that's going to result in is a big increase in the dry length of those intermittent headwater streams. So these are different climate scenarios for each graph, and then each year kind of shows like the projection. And then as it gets taller, that's like an increase in dry stream length. So essentially, we estimate that we're going to lose around 5,000 kilometers just within the next 3 or 4 decades. And then there's also different ways in how logging is treated in headwater streams. So these are kind of the buffers that we're all very used to. So when you do timber harvest, if the stream is fish buried, then you're kind of required to leave this buffer of trees along the water, and that helps prevent more sun from reaching it and increasing the temperature of the stream. Sedimentation can happen from logging, and that's where you get a lot of dirt and fine particles. It's going to kind of clog the pores and remove oxygen from the stream. So there's a lot of things that can happen, and so we have these riparian buffers kind of built into the law so that we don't get a big impact from logging. However, that only applies to fish-bearing streams, and the headwater streams in which torrents live don't have fish. And so they're not protected in the same way that some larger streams are. And so it's thought that this kind of joint threat of climate change and increased logging, especially now, into the future could potentially be difficult for torrent salamanders. So in terms of conservation and thinking about, well, how do we kind of protect this species into the future? Kind of the most important question now is just, how are torrents doing? So it's a lot of different things. So like, how big are the populations? Are populations declining? Are they healthy? Are some populations maybe more locally adapted to different conditions? And it's hard to get at that by traditional methods. So there are some folks at the USGS that are doing some studies on the ground. But generally speaking, in terms of counting numbers, that can be really difficult because these Cascade torrents are kind of known to be very difficult to find, especially seasonally. And in terms of kind of measuring the health, that's gonna, traditional methods rely on finding the same ones over and over and over, kind of tracking that over time. So there is kind of another way to do that, and that's through genetics. And essentially, genetic diversity within a population means a lot of different things. It's essentially the raw template upon which all subsequent higher levels of diversity are formed. And that's very much tied to things like adaptive capacity. So there's more genetic diversity in a population, then if a disease were to come through, or say a fire came through or something like that, there's a higher chance that there's going to be some individuals that have the genetic, bank to handle that. And there are some studies that have shown that after fires, I think it was with frogs in Washington, only frogs that specifically possess the genes for heat shock coating protein survive that fire. So that's kind of an example of that. So generally speaking, large populations have more diversity, but we don't really know how large they are, and we don't really know how connected they are either. And that's kind of because of how big their range is. So for Cascade torrents, it's just south of Mount Rainier up until about Oak Ridge, which is just southeast of Eugene, Oregon. And there's a lot of different barriers throughout that. Most notably, the Columbia River, which is very large and a little bit too big for a salamander like a torrent to cross. So with that and kind of all the different habitats, it's possible that there's different populations and different kind of genetic adaptations. But there's only been one genetic study of the species, and that was in 1987, and it only had a handful of them. So we wanted to kind of expand upon that. So we went and did a range-wide study, pulling 144 individuals across 27 sites in both Oregon and Washington. And we collected just a centimeter of tail tip from the field and stored that in ethanol. And the tails do grow back. It's one of the cool things about salamanders is they all have some regeneration, and torrents seem to have it pretty well. They're also kind of adapted to losing the tips of the tail. So generally speaking, like when you cut the tip of the tail, it'll kind of wag, just like a skink. So it's kind of, they already have a built-in defense mechanism for it. And then from that, we wanted to not use allozymes like the old study, but use single nucleotide polymorphisms. And essentially the way that works is, say you have 3 different individuals just at one spot, one nucleotide, you can see differences between them. And if you get a lot of those, you can get a lot of information about how related one is to another, and depending on where it is, how functionally different they are. But of course, we're drinking this with torrents. And essentially the way you kind of generate that data is by taking all of the DNA from an individual, cutting it into these slices, and then from each slice, you just pull one nucleotide, and then essentially you just have this big bank of thousands of SNPs that you can do for a lot of different analyses. But there's not a lot of studies like that with salamanders, and that's for an interesting reason. So if you think about something like a deer, its genome size is 3 gigabases. And that's pretty much standard for most vertebrates, and humans are very similar. And if you compare that to the torrent salamander, it's a 70 gigabase genome, so orders of magnitude larger. It's not even the biggest salamander genome. I think the biggest is like 124 or something like that for mud puppies. Absolutely massive genomes. And the reason why that's a problem for genomic studies is because, we were talking about how we needed to cut them into slices and pull these markers. Well, you can imagine that, say you cut, 4 slices from this deer genome, it's a high chance that you're going to be able to get similar ones across individuals. But it's going to take a lot more cuts among such a big genome in order to do that sound. So we kind of developed some methods to deal with that. So briefly, essentially what we did for each individual is we took the tail tip and then we subdivided that tail tip into 3 tissue samples. And then for each tissue sample, we did duplicate DNA extractions. And then we did that through the magic of RADseq, which is where we cut the little slices and do loci. Then we end up with all of these reads per individual, which we then compared to individuals from the same site, and then compared those sites to other sites. So kind of with this raw SNP data set, this is actually 144 individuals changed made it. It's millions of reads. And then we ran it through filtering. And this is a very small clip art that took me 1 second. And this process has taken about 2 years. Because as you can imagine, for millions of reads, it's pretty difficult to kind of filter out the stuff that isn't informative. But with this data set, that equaled 140 individuals and about 8,000 SNPs, which is pretty good. And then we were able to then jump into analysis. And so I can show you some of the results that we found for what kind of genetic structure looks like just within Cascade Torrents. So this is a PCA plot. So essentially what it does is it takes all of the diversity that's present in those SNPs and it distills it into what is the thing that explains the most to amount of the variety. So on the x-axis, it says PC1, so about 20% of the variation, which is pretty high for this kind of analysis, is basically explained by if an individual comes from these 4 sites on the right, which are in Northwest Washington, or the rest of the range. And when we get PC2 and PC3, we can see that kind of breaks a bit. But you can see that this kind of analysis doesn't take geographic location into account. And so this is essentially just based on the SNPs alone, saying if you see things in a group, they're more related to each other than things that are in a different group. And so the green is the western side of the Columbia River Gorge on the Oregon side. And what was really weird, and I had to go through the data a million times to make sure it wasn't my fault, and I messed up or something, is that the stuff on the east side of the gorge on the Oregon side does not line up with that, which is really interesting. So we took those weird divergent sites out that were the Northwest Washington and ran it again. And yeah, we found again that the stuff on the west side of the gorge kind of lines up pretty closely with Mount Hood. And then you get this big kind of clump that's all of Oregon, south of Mount Hood. And then you get the stuff in Washington, and again, the stuff on the east side of the gorge on the Oregon side. And looking at PC3 and PC2, still get that same pattern where now the very bottom of that are the sites on the eastern side of the gorge, and they really kind of stick apart from really most everything else. So then we did a more model-based approach. So this is using fast structure, which is, long story short, it's a Bayesian model. So it does a lot of different things to calculate how related something is, and then so tries to estimate how many ancestral populations existed within the species. And it said that there were 6 populations, and that again, it did list those 3 sites on the west side of the gorge very separately from the stuff on the east side of the gorge, which is much more closely related to stuff on the Washington side, in the kind of Trout Creek, Panther Creek area. Which is very unusual. So that kind of begs the question, what's really going on here? One of my friends, when I was kind of poring over this and trying to figure out what was going on, it was very funny. And she was like, well, maybe they just at night, they just go and cross the Bridge of the Gods, because the Bridge of the Gods is right there in the center of kind of this weird relationship. And obviously that's pretty silly. But the Bridge of the Gods was named after this landslide. So we know that this happened in the 1400s. And while that landslide was there, the Oregon and Washington sides were connected. So it's absolutely possible that salamanders could have been crossing over that the whole time. So that could be an explanation for the pattern that we see. Is that, when that landslide happened, the downstream part, or sorry, the upstream part of the gorge essentially turned into a giant lake, because as much as that water was kind of coming up against this new barrier, it flooded part of the gorge. So it's possible that it kind of killed off the torrents that were already there, and then they were kind of recolonized by stuff from Washington. But there is another kind of potential hypothesis, and that's that maybe they were there and the river moved around them. So we do know that the Columbia River has moved. If you think about 3.5 million years ago, it moved northwards from its original kind of path. So it's possible that where it moved, it kind of separated some that were more related to Washington. So maybe this happened kind of shortly after colonization. We really kind of don't know. But there are some ways that we can kind of test that. So we know that the Columbia River moved northwards about 3.5 million years ago. And then the Bridge of the Gods landslide occurred somewhere in the 1400s. And it is, after talking to geologists, possible that there may have been multiple of these events, because the Columbia River Gorge is a very active place in terms of landslides. So it's possible that maybe things happened in the past. And so one of the ways that we're going to do that is kind of sample these additional spots to try to figure out exactly where that break lies. But there is another way that we can also test that, and that's by using phylogenetic trees. So this is a tree of all of Cascade. And if for those that haven't seen this before, essentially what a phylogenetic tree does, this is a family tree. So each individual has its own separate branch. And then where you get kind of these clumpings, it's where things are very related. So in this, you can see, everything in the blue on the top there is this kind of southern Oregon part. The west side of the gorge is the green. The pink is Mount Hood. The east side of the gorge is down over here in the purple, and then Washington is over here. So you can even see in this that, generally with the way that these work is the longer the branch, the greater the time between. And so it looks like that kind of divergence between the stuff on the west and east side of the gorge is very, very old. So it might be more likely that the river moving is the reason, but we're not sure. Torrents are very old. They're about 20 million years old, which is shocking because it means they've been here, before the Missoula floods, before the Missoula floods. They've gone through, they were here before St. Helens. I think St. Helens is only like 80,000 years ago. So it's totally possible that that's the reason why. And the way we're gonna actually, I guess, scientifically deduce which of those is the case, as it's possible that it might be both, is by using a dated tree. So, we know that they're about 20 million years old. So what we did is add a new data set, and that's called ultra conserved elements. So with SNPs, we're getting stuff randomly along the genome, but not all parts of the genome kind of mutate or evolve at the same speed. There are some things like, the genes that code your immunology are not going to get a bunch of mutations because you'll die. So there are these parts of the genome called ultra conserved elements that evolve very, very slowly, and it allows for a look much back further into time. And so this is a tree we've made with that data set, where you can see kind of torrents are this large grouping, and we've included all species of torrents in there. And each of the other branches are representatives of the other salamander families, with the oldest being hellbenders and Japanese giants all the way to the left, and then the sirens on the bottom, and then kind of you can see how the lungless salamanders are actually the closest relatives to the torrents out of this grouping. And then you can see a lot of the Pacific giants, or the VITE, and the AMMA, or the mole salamanders. And so using these ultra conserved elements, we're hoping to essentially put dates on divergences. and then use that with the SNP data set to say, okay, well, like how old is this? And when did this break occur? That's kind of what I have for you. And then also I'll just do a little plug for our bilingual English and Spanish book that we do events at. Actually, next on Saturday, May 4th, we're going to do something at Tryon Creek State Park, which should be very fun. We've done it in the past and it's a good time. And with that, yeah, if there's any questions, take them, whatever.
Perfect. Thank you so, so much, Chris. Now we are going to step into our Q&A portion. So again, if you have any questions for our speakers, go ahead and add those to the Q&A right now, and we'll get started. Bear with me. Oh gosh. There it is. Okay. All right, so Christopher, one of the first questions that I have for you is, when we were doing the intros, we got to know a little bit about how Kenzie fell in love with herps. So I'm really curious, for you, what was it about salamanders and what was it specifically about torrent salamanders that made you want to dedicate your PhD to studying them?
Yeah, that's a good question. Well, I don't know. Ever since I was a little kid, I kind of always liked animals. I guess I didn't know for the longest time that it was a job, and then as soon as I found out it was a job, I was like, ooh, and so I tried to jump on that. But I've always really enjoyed finding amphibians. I think they have really interesting lives. And as far as like torrents specifically, I didn't like kind of choose that. It was kind of fell into my lap. Like I got hired on to do other chapter of my thesis, which is environmental DNA work and doing stuff with torrents. And then it just kind of lined up in the perfect time where I got funding from this foundation and I could kind of do what I wanted with it, and then found out that these torrents were going to be listed. And the way the Fish and Wildlife Service works is when they list things, they put kind of dots on where specific populations are, and so it just kind of worked out perfectly. But now that I've been working on them a lot, I really like them. They're also really sparkly, so it's cool.
Thank you so much, Chris. Okay, so Kenzie, next question is for you. Where do you bring students in the field to see salamanders?
I'm really lucky that our programs that we already had established at Beacon Rock State Park, that is an excellent spot to go looking for specifically the western redback salamander, as well as there's some large mountain salamander if you're lucky in that area. So that's a great place to take them. But if you're native to Portland, Oaks Bottom Wildlife Refuge is a place I like to take kids or my friends in my free time, because there is hundreds to thousands of salamanders there, and you're guaranteed to see them if you're looking in the right spots on the great day.
Okay, and this question is for the both of you. What would you consider the most threatened of the salamanders in the gorge to be, and which ones are doing the best?
I can say that duns are like the tanks of the amphibian world. Like you just find them everywhere. They're also bigger than the hiculum or the redbacks, so they kind of bully them out of like the best spots. There's some interesting papers about that. So yeah, I would say duns are probably doing the best. And then as far as the worst, I think that's a tricky question. I don't think we thought that large mountains were hit pretty hard by the fire. But I was part of some Forest Service research that went through the fire stuff and kind of did surveys, and it looked like, not only did we find kind of larvae, but we also found adults, so it looks like they're okay. Just with the talus being so deep, it's such a refuge. I think there was like a pica study that found that even down deep in the talus during the fire, like the temperature really barely shifted at all. It's like such good refuge. So yeah.
Yeah, I'll add on to the rough skin newt is doing very well as well. and you can find quite a few of them still pretty much anywhere in the Pacific Northwest. That there's quite a few big breeding ponds out there near Mount Hood, near up north of Vancouver, Lucia Falls. Very prolific breeders and doing good overall. But one thing I want to note is amphibians all around the world are at risk due to climate change, due to habitat loss. So that's something to keep in mind with amphibian conservation is it's a very steep fight we have up ahead for amphibian conservation.
And Kenzie, with that topic in hand, one of our questions by Betsy wants to know if either of you have seen examples of the amphibian populations decreasing in the gorge or Northwest in general.
Yeah, I'm not familiar on a lot of the studies for different species that have como se dice, like very specific data to that. But I will say when I was getting samples for the torrents, and I don't want to like speak to other people to research, but I think it backs me up on it. But you know, I was going to historic sites. So like these are sites that like we know they're there, or at least they used to be there. And it was really hard to find them. A lot of these sites like just dry up, you know. So I think that generally speaking, there's evidence that the torrent salamanders specifically are declining in their range, but I can't speak to other ones.
Yeah, it's hard to know because, like, personal experience wise, you know, around my neighborhood, just barely outside of the Columbia Gorge, like anywhere that's been severely developed where wetlands have been taken away, you're going to see less amphibians. But in terms of like in the gorge, it's hard to know unless you're looking at the research and the numbers. And, you know, there's not studies being done on every single species all of the time. So, yeah, it's hard to know for sure.
And you two were speaking a bit about the talus field, and for example, Chris, when you mentioned the pika and the talus and salamanders being found in there. Can subterranean salamanders though dig their own holes and tunnels, or do you usually see like if there are other species that have their own dug their own tunnels or areas like talus fields, do they just tend to already find holes that are already created? Can they make their own?
Yeah, I can kind of again I'm not like can't speak to all species, but I know that for some of the larger mole salamanders, they're generally kind of known to use rodent burrows. And there's some really interesting evidence of them getting along with them. Actually, there's and I'll just kind of just super briefly say, like one of our salamanders, the clouded salamanders, there's been some really cool work with trail cams up in the top of trees where they have what essentially looks to be a very good relationship with red tree voles, where they have them on camera. They'll live in the red tree vole nest and eat all the insects that are in the nest. When the red tree voles come up to them, they just kind of look at each other and then keep going on their way. So I think, you know, they're very used to dealing with mammals or small ones.
Yeah, I'll add it just depends on the species, but a lot of salamanders do use the burrows, as Christopher was saying. But some are able to dig themselves down too, as well.
All right, this is more of a general question. Why do some salamanders have grooves? What is the purpose of the grooves?
To increase surface area on their skin to allow better moisture retention is usually the case. But Christopher, do you want to add any more than that?
No, I didn't know that. So I'm glad you said it. I was like, I don't know what they're for actually. So thank you, Kenzie. I learned something too.
I had a student ask me last week, and so I did some research into it. So I'm very glad you asked that because I was just asked this week.
Do either of you have anything more to say on how the Eagle Creek fire affected gorge salamanders?
Yeah, just like I mentioned, you know, we for the large mountain salamander specifically, like for the Forest Service, we went to some spots that had burned, some fires that had not. And it, you know, while we were kind of less likely to find them, it doesn't mean that they weren't there. That often is just kind of a result of how different the habitats are after. But it looks as if it was fine for that species. And then from not in the Eagle Creek, but some of the sites that I've had for Cascade Torrents, like near like Detroit Lake, and kind of those areas are in fire zones. And they're like very difficult to find, but they are there because they're able to kind of use that subterranean zone. And so I think torrents are kind of almost very similar to large mountains, and a large mountain have those kind of talus that they can go down into. And then as long as the habitat supports it, that torrents have that kind of cobble that they can go underground and be where the water is, kind of weather the storm, so to speak.
Yeah, and one thing to add too is that like when the fire was burning hot, any salamanders that were not deep underground, they would not have survived that. They can't move away quickly enough. But a lot of areas have had salamanders returning, and Oregon State University has been doing research on how amphibians come back to areas after fires. Christopher, do you know when that paper is being published?
I think the Page Prentice one is published. And then as someone else, I'm not sure who, I just heard about it from a community member, is doing stuff now with Larch Mountain too. So I think there's going to be a lot, now that all the fires are such a thing, we're going to see, I think, there's a lot more research being done there. So there should be more coming out soon too.
Thank you so much. Okay, Chris, this is a question specifically about torrent salamanders. Why is it important to know the genetic information for each of the torrent salamander populations?
Yeah, so it's not so much to know them for each. It's just that we didn't know where the populations existed. And what I mean by that, I guess, is how I'm trying to think of the fastest way to answer this without going into that. So essentially with the kind of like genetic populations, you can have something where it's an isolation by distance. And so like say, at just the farther away you get from an individual, the less related it's going to be. But then you can have kind of a different thing, which is what we see in torrents, where you have these very distinct groups where gene flow is only happening within them. And so when you have things like that, then it's just kind of important to know that, like, for whatever reason, something is happening that there's not gene flow. Like the big question, I think, for us is like, why are things east of Bridge of the Gods, apparently there's no gene flow at all with things from west of the Gods, and there's no like barrier there. And so that could mean that there's kind of a different adaptation that has occurred. Certainly, I think that's one of the big things. With this, because the genomes are so big, we don't have a lot of the functional information. But with things like birds that are a little easier to study, this data has been used to really pinpoint specific things, like saying, okay, well, this bird that lives on the south is actually a lot more tolerant to living in drought conditions to something on the north. And, unfortunately, eventually, like, things don't do well and you have to have breeding programs. And that's where this information becomes really useful, is when you're trying to release individuals back into the wild. If you're not releasing individuals with the genetic material that's made for that environment, you're kind of setting yourself up for failure. So that's kind of a big thing that we keep in mind.
Thank you so much. Okay, so with lungless salamanders, they absorb oxygen through their skin, right? Is it possible for them to not get enough oxygen?
Yes, if they're not moist enough. Them being moist is a tool that keeps them oxygen flowing through their skin.
What does it mean to be fully metamorphosized?
That just means that they go through the full process. So I'll compare it to frogs, since they have a more dramatic metamorphosis. They start off as a tadpole, they slowly grow their back legs, their front legs, they absorb their tail, and then they're hopping on their way. So for salamanders, it's a little bit less dramatic. They look pretty similar to adults would. They have gills coming out of their face. They have 2 legs, right, Christopher, when they hatch, and then they slowly get their gills back into their head. They get all their legs, and then if they're terrestrial, they'll go out and on their way. Or if they're aquatic, they will remain in the aquatic format. Christopher, do you want to add anything?
No, not a lot. I mean, that pretty much, I think one of the things that's so kind of interesting is that, it's 2025 and we still kind of don't understand why some individuals metamorphose and others don't. And that's kind of an active area of research with the Pacific giants. Like, why is it in some streams they all stay in the water and their entire lives, and then in others they decide to metamorphose and leave the land? And it's kind of, we thought it had something to do with resources, but it looks like it's actually pretty complicated. So it's kind of cool.
Okay, so for our viewers tonight, if there are 2 or 3 things that you want them to walk away from this webinar in terms of the benefits of salamanders to the environment, what would you say?
Amphibians in general are an indicator species of the environment, so they're a great way to know how an environment is doing in general. If there's frogs, salamanders in an environment, the environment is likely to be doing okay to healthy, because they are able to absorb through their skin moisture, oxygen, etc. If there's pollution, they are very susceptible to it. They're not going to do well in polluted streams. There's always exceptions. There's some amphibian superheroes that are able to survive in pretty much anything in pool water, but for the most part, they're great indicators of the environment, and they're important for our food webs. I love them. I hope you all love them too.
Yeah, I think that part about being important to food webs is really important. I think a lot of people underestimate like how many of them there are. I think there have been some studies on the East Coast that have found that if you were to just take biomass, so just raw weight, the weight of salamanders is more than the weight of all the deer in the forest. So we're talking about things that are like, you might not see them, but they're there a lot. In terms of, each group is going to be a little different, but for things like the lungless salamanders, they're actually really, really important in maintaining decomposition of the forest floor. They're eating things like springtails and mites and other kind of more soil microorganisms. And so they're really important to keeping the decomposition rates going. Again, they're really important food sources for, much in the same way that when salmon go upstream and die, they become really important sources of nutrients. For headwater streams, these are the source of nutrients.
Thank you so much. We have a couple more questions. I've got some really good ones. Okay, next one I want to ask is, do fungi pose a similar threat to salamanders as they do to frogs?
Yes. Yeah, there's a group of folks. It's the Jonah Peele Scott Lab at Washington State University that it's kind of their whole area of research, because as the way Kittredge just completely wiped out frog populations is really well known. And the one that we're kind of worried about for salamanders, I think it's called B-sal. And so one of the things that they're doing with their research is, finding out how sensitive are different, groups. So like newts, they're very sensitive. Clouded salamanders, maybe not so much. And so they're already kind of, the general thought is that it is a matter of when, not if, B-sal comes to the US. And they do expect to see some large die-offs, and so I think they're trying to get ahead of that right now. But yeah, that's one of the biggest, and that's actually why you can't buy pet salamanders in the store from out of country, because all external salamanders are strictly forbidden, so that way B-sal doesn't come to the US. So yeah, it's a very big threat.
Yeah, and I'll add, I worked in that lab and under lab, and we were directly exposing California newts, rough-skinned newts, Pacific tree frogs, the newts to the B-sal, but it causes lesions in their body that are often fatal. It's really nasty, it's really painful, it's really awful, and I am sad for the day that it shows up in the environment.
Thank you. Okay, well, with that, to follow up, what can we do as individuals to support salamander conservation?
Big one is support environmental nonprofits, shameless plug to Friends of Columbia Gorge, or any other that promote land conservation, habitat protection, organizations such as ARC, Amphibian Reptile Conservancy, which I see Christopher's wearing a shirt from them. So lots of great orgs doing work for conservation. So direct financial support is a great way. Volunteering, there's plenty of citizen science programs where you can go out and get data, since there's not enough biologists and field techs to get the amount of data that we need. Learning what you can, leaving animals in the environment. There's so many things you can do, but the biggest one is supporting the nonprofits and the organizations that are doing the work to protect salamanders.
Yeah, definitely. The nonprofits are so important, not just amphibians, but and I think in the years to come they'll be even more important than they have been in the past. Nature Conservancy is like another great organization, and there's a lot of them. And I will say too, when I was like when I found out this was a job and I was trying to get like experience to get into um college or whatever, like I did a lot of volunteering with Oregon Metro because I used to live in Gresham. And Oregon Metro does the amphibian egg mass surveys, which are a lot of fun. You get to go out with biologists and do that at active restoration sites. So honestly, like we're very lucky in the Pacific Northwest to live in an area where there's so much going on, because it's not always like that in different parts of the country.
Perfect. And Christopher, I have one last question specifically about torrent salamanders. Why are torrent salamanders specialized to live in headwaters of river systems? Why have they adapted to live in these habitats specifically?
Yeah, that's a great question. And I don't know. I mean, we don't really know. We think that a big reason why they kind of don't move down is because there's predators. So that's one of the big kind of risks to torrent salamanders as climate change happens and kind of the upper reaches of headwaters become unsuitable. If they were to just move down into some of the lower stream, for one, like the oxygen and the temperature might not be good enough, but there's things like salmonids that would probably eat them. We think they are toxic to Pacific giants at least, which would make sense given their coloration. But yeah, I mean that's such a hard question. It's like why? I don't know. At some point in the past, like they found a spot they liked and they stuck with it.
Gotcha. Okay, I've got 3 questions left. Okay, what is a good source of photos for salamanders, especially the one with the frilly parts on the head / neck? Christina thinks they're so beautiful and that it'd be fun to paint one.
Shameless plug, Christopher's Instagram. I don't know if you want to give your Instagram handle. He has amazing photos on his page. Also the Fish and Wildlife websites, they have very clear, correct, identified photos for you to get learning more.
Yeah. I don't know if I can do that in the chat or whatever. But I guess I can. Yeah. I'll throw it in there. But yeah, I mean, it's one of my side hobbies is photography. It's actually great because when you give talks, it just like really brings people in. And there's nothing as boring as a talk. I feel like every paper and every talk, it's like should be like the law that there's at least one photo of the species, to like draw you in. But also I think there are some other things. I think ARC has some. And there's a lot of kind of open access. Also, iNaturalist is a good source of photos. Because when you like upload to iNaturalist, like you tell them what the license is. So that's a good one. And I'm half Mexican and most of my family lives in Mexico City. And there, axolotls like ajolotes are like huge culturally, and so there's a ton of art. And so I would, it's even on the money. Like that's how big they are. So I think there's a lot of good examples of that kind of art from Mexico.
Thank you. That's awesome. Okay, so Christopher, you did mention your hikes with Tryon Creek, and that one's coming up. So we have the hikes with Tryon Creek. Do you know of other orgs, or do you partner with other orgs or institutions to lead herping field trips in Portland or the greater Portland area?
I personally don't, and that is more like because we have the children's book, so we do kind of like readings of that, and then just do like short walks, which again has been cool because there are Pacific giants and Titinas and a lot of things at Tryon Creek, which is the biggest state park in the metro area in the U.S., which is a cool little fact. But I actually don't do a lot of that. There's not a lot of that in the Pacific Northwest. I mean, the salamander world is very small, so I have a lot of friends in the Southeast, and they're all right now doing a huge thing in the Smokies where like they're all leading salamander walks and stuff. But I don't really know of any of that here. Kenzie, I mean, do you know of anything like that?
Yeah, Friends of Oaks Bottom puts them on every now and then. They have field trips if you're looking for a school group. Oaks Bottom has quite a few educators on staff, so there's quite a bit of access. And the big thing about Oaks Bottom is the salamanders are right off the trail. Because one thing that's really important when leading groups or herping in general is to not cause harm to the environment. If you're taking 20 people out off trail to go look for them, that can be quite damaging. And I saw another question about Friends of Columbia Gorge leading them, which I led one last year in June, but it was a little too dry and hot the day we went out, so we couldn't find anything. But stay tuned for our outdoor programs newsletter to hear about any opportunities in the future, because I would love to host more salamander walks, reptile walks, etc. All things herp for me.
All right, last question is there anything that you would like to plug or a way that the viewers can follow your journeys as a herpetologist? Christopher, you mentioned a book. Could you tell us again about the book, a little bit more about the book? And yeah, just in general, you know, as you finish up your PhD, like how can we keep in touch with both of y'all? Just, yeah, how do we know more?
Yeah, I'm on Instagram. It's just Christopher.cousins.wildlife. And though I don't post very often, and most of the time it's just stories of my corgi, but that's fine. Yeah, and the book is kind of more of where I think we do a lot of our outreach. And the site for that is noestadabooks.com. It's free for educators, and most of the time we just go out and do readings. And one of the authors, Isabel, like is social scientist, so we actually have like lesson plans and stuff. We've done it at dual immersion schools and stuff. But I would say probably just instead, though I haven't been very active. I don't know, everyone has like this fancy website and stuff, and I just like have not been able to do I don't know.
Yeah, for me, I'd say keep watching our Instagram page for wildlife features. I've been seen a few times talking about some of the salamanders in the gorge on there. Stay tuned with our outdoor programs list to see if I'm leading any. And then outside of my work with friends, if you would like to learn way more, you could take my Biology 145 course at Clark College. It won't be available again until next spring because we're currently in the middle of a term, but you will learn so much in that class if you really want to do a deep dive.
All right, everyone. Well, thank you so much for watching and participating with us. These were some awesome questions and a great webinar, just getting to know more about salamanders in the gorge. So before you go, you know, if you liked what you watched tonight, this is just a glimpse of many more that we are going to host throughout the upcoming year. So if you enjoyed this webinar and want to support our work, consider donating to Friends by visiting gorgefriends.org/donate. And then in general, just thank you so much for your support. Keep an eye out for email, social media posts, as Kenzie said, and just thank you so much to our speakers. Everyone have a great night. Enjoy your spring.
Thanks, everyone.
Thank
Happy
you so much,
herping.
speakers. Happy herping. Bye, everyone.